welcome Dr. Miraz Rahman to this, the next of our Spotlight conversations. Why don't you start by just telling us a little bit about your own background and how long you've been at King's?
So my name is Dr. Miraz Rahman. I'm working as a reader in medicinal chemistry at Institute of Pharmaceutical Science. And I've joined King's in May 2012. So it's been about 8 years I've been at King's.
And tell us your main areas of interest and research. What are they?
So I will identify myself as a drug discovery scientist. I'm really interested in antimicrobial research, identifying new therapeutics against both bacterial and fungal pathogen. That is the key area where we are working with Public Health England and some other partners. I would say that we are interested in identifying both chemical tools to understand the disease, at the same time identifying new small molecule therapeutics against different pathogens, mainly public health pathogens, but in some cases emerging pathogens like Candida auris, which is in many cases is considered as one of the most concerning pathogens because it has got a mortality rate of about 65%.
So we are having a conversation today mainly because of the work you're undertaking in relation to coronavirus and SARS-CoV-2 in particular, which as a virus isn't one of the pathogens that you typically have focused on. So tell us a little bit about what has motivated you to get involved in at least seeking to address some aspects of this COVID-19 pandemic.
So our interest in these SARS-CoV-2 started when we started to see the devastating effect it was having in Wuhan, China. And as I have said that I work with Public Health England in our other pathogenic targets like bacteria and fungi. So we were interested into seeing that whether we could do something in finding new agents that could be deployed against these pathogens. But at that time, there was no information available about targets that we can target to design small molecules. So we had some discussions with Public Health England. They have got a very good viral group. They run these both in vitro and in vivo models, or mainly for pandemic influenza model. So when we discussed this and they inquired whether we would be able to collaborate with them. And at that time, we found that both Diamond Light Source and Seattle Structural Genomics Consortia, they published cryo-EM structure of both bacterial spike protein, the viral spike protein, which is very important for gaining entry inside the cell, as well as the main protease. So when we saw the structures, then we knew that we can start to work with our techniques, where we used very advanced computational techniques to identify either small molecules where we can start from scratch, or we have been hugely successful in the past in different drug repurposing projects. So we thought we will start a project where we will have a short-term goal, where we'll try to identify drugs that can be very quickly deployed to clinic for clinical trial. At the same time, we will start a long-term project where we can screen large compound libraries, design new drugs against these viral targets.
And that dual approach, I guess, is recognizing there's an immediate need for some therapeutic intervention in this disorder that's having such devastating effect, but also recognizing that optimally there might be a much longer lead time to some other targets that might emerge because, of course, typically the lead time for entirely novel therapies can and often is very extended.
Yeah, that's exactly right. Because the pandemic is spreading so fast and having no treatment options available, we need something into the clinic as quickly as possible. And if we can identify drugs, which we know that FDA or EMA approved with known safety profile, and if they specifically target different stages of viral infection, then we can have an informed option to initiate clinical trial. Because in many cases, the clinical trial has already started where we do not know enough about these drugs, whether they are actively working on any stages of viral infection, like hydroxychloroquine. This has been used in the past, but we precisely do not know just raising the pH of the endosome is enough to prevent the viral earlier stage of infection. And also, long term, we need drugs because these probably will stay with us for a longer time, and we might see again in the future. So we need drugs specifically targeted against these pathogens. So the long term approach is equally important.
Indeed. And I know you'll be talking more about the progress you're making with your research in one of our research fora. But to finish, and for now, can I just ask your thoughts on anything you felt has been outstanding in regard of King's College London's response to this pandemic?
I think the way the King's community has come together in addressing this challenge. King's always responds to global challenges, and in many cases we cannot do lab research, but in my research project, what we found that we were able to do some lab research. The college was very helpful in giving us access to the lab. I found that everyone, including people working in purchasing in stores, they became like a research component. The way they arrange the delivery of samples to my home, from here to Public Health England, it's been really fantastic. It looks like we're working as a team. And this belongingness, sense of belongingness, everyone wants to contribute. They want to feel that they're doing something in addressing this challenge. That has been fantastic. And I must say that I also found the willingness of partners like Public Health England. I'm working with IQVIA for clinical trial guidance. They have been fantastic. Everyone is really trying to help each other.
Well, Dr. Miraz Rahman, may I thank you for your time and talking with us. May I thank you and your team for all the work they're undertaking. Thank you.
Thank you very much. Thanks for having me.